Great. So thank you, Laura, for this amazing presentation. And yeah, well, yes, I'm Guillermo, together with my colleague Jan. We did this tag team presentation, which is like the main theme is to talk about how protein databases influence our analysis in paleoproteomics. So yeah, perfect. So the first thing we need to think about is why are protein databases so important? So for those of you who are not really into the technique of shotgun proteomics, this question might seem not really with a lot of sense, but I promise it really makes sense because databases are not all only important, but are essential for this technique. So basically, just as a reminder, what we do for shotgun proteomics is to measure each part of all the peptides we have in our sample to build what we call an experimental mass over target spectrum. So we have like a measure of the mass of each part of the peptide. And for knowing the sequence of this spectrum, what we do is compare it with a set of, or a library of theoretical spectrum built in silico through a protein database. So basically what we do is to input a protein database into this type of software, and this software build this library of theoretical spectrums, and then we try to match the experimental spectrums with the theoretical spectrums. And if two spectrums are similar, then we can infer the sequence of the experimental spectrum through the sequence that was used to build the theoretical spectrum. And there are many softwares that are able to do this. One of them is MassQuant, probably the most widely used, but there are, of course, many others. So we thought that there are like three main features that protein databases should have for this process to happen correctly. First of all, they need to be non-redundant. So to avoid extra comparisons, also like if you include two records with the same sequence, softwares can mess things a bit. Protein databases also need to be parsable, like probably in a multi-FASTA format. This is like the format that is used for many of the softwares. And protein databases should also be diverse but concrete. And this might seem counterintuitive, but I promise it is not. And I will stop here a bit and try to explain this. So here I present you some scenarios of what can happen in terms of the peptides we have in our sample, like the real peptides we want to identify, and the peptides that we have in our database. Of course, if a peptide is not included in our database, we will not identify it. So here are this first scenario when our database is rather small and most of the peptides remain unidentified because there's not a good overlap. Then we have this second scenario when our database is huge, and of course, many of the real peptides are represented in this database, but this also leads to a high risk of false positive identifications. And this intuitively means that just by chance, there's a probability that we are identifying a peptide because of database is huge, that is not really there. And this also leads to a really long execution time, and this is not acceptable in many ways. And there is this third scenario, we call it the good enough scenario. So basically, this means that we have a rather small database or with a relatively standard size. Most of the peptides in our sample are represented in that database, so we will identify most of them. And this is what we really want to achieve. And there's no way to know in advance which will be like the desired size of our database or which proteins we should include. But here we want to give you this intuition. Yeah, so the thing is that those databases were typically done manually or like reused from other papers. But as you may have seen by like in this conference, there are many more species being analyzed day by day, and the increase on paleoproteomics has been increasing. The interest, sorry, has been increasing. And so we thought that it would be great to have a pipeline to automatically build databases through a certain conditions. So it's like easy and more replicable to do this type of analysis. And then we created ProteoParc that I already presented the logo in the last slide. And this is basically how ProteoParc works. So you just need to input two things. The first one is a tax ID, which is a number that indicates a clade of the evolutionary tree. It can be mammals or it can be Homo sapiens. So from species to even classes, orders, phylums, whatever. And then you can input a list of genes, which is optional, but it will be desirable to restrict your search. So then the pipeline will try to download from UniParc, which is an archive that includes proteins from RefSeq, UniProt, Ensemble, like the most common use protein sequence repositories. So the pipeline will download proteins from there under this combination of species and genes. Then it processes somehow the database, like removing the redundancy if it finds like two records that have been downloaded twice, or like records that are a fragment of another record. So there's also redundant information there. And it also, there's an option for aligning the database. So you can like easily input the multiple sequence alignment in a multiple sequence alignment visualizer to see if there's some records that are really not good quality and you can get rid of them from your database. And finally, it produces some metadata files to see really what's in your database, so you can, again, easily see which species have you downloaded, which proteins have you downloaded, and so on and so forth. And then it's the turn of Jan to explain like what will we be doing with these databases.
Right. Yeah. So let's see if this seamless transition works as intended. So with this brilliant tool, of course, we wanted to put it to the test. The main question being, do these narrow targeted databases actually result in more protein and PSM matches than these wide databases that we've been using before? So to test this out, we used we took samples from 3 different material types: bone, enamel, and dietary complex mixtures, specifically in this case, ceramic residues and calcified deposits. For each of these material types, we took one publication and just took a small selection of 5 to 4 samples each. We tried to ensure some measure of taxonomic diversity. And then for each of these materials, we created a database using ProteoParc. So the way we did this is we did a quick literature review, trying to find both modern proteomics as well as archaeological proteomic papers. And we extracted all the proteins identified in those. In the case of bone, we limited it to all proteins for which were mentioned in 4 of 13 publications, in order to, because the modern proteomic papers on bone, they find thousands of bone proteins. But we, in archaeological samples, often find a much smaller selection. So to ensure that we only took the proteins that you are actually likely to find, we had this filter of needing to be present in 4 publications. So we took all these proteins, extracted their gene names, and fed them through ProteoParc and downloaded the mammalian homologs for these 104 genes. Now, for the dietary mixtures, we just downloaded the protein IDs that were actually observed in the paper, whereas for enamel, we didn't download the genes to the level of mammalia, but rather on family level for the particular samples. The reason why for enamel we decided to be more specific is because with an enamel sample, you have more of a pre-notion of what taxonomic identity the sample will have, just based on its morphology. Whereas with very fragmented bones, this is of course more difficult. Then you can see the final database size on the bottom. So then we ran these databases through MaxQuant with against the previously mentioned samples. The parameters for MaxQuant are fairly standard. For the enamel and dietary samples, we did an unspecific digestion mode, whereas for the bone samples, we did a triptych digestion in MaxQuant. Now, the one big difference is that we ran these samples on a high-performance cluster. So as you can see, we have quite a lot of memory, and we use 4 CPUs. Well, so if you're running MaxQuant on just your laptop or a desktop, The running times that I'll be presenting later will probably be quite a bit faster than on your own local machine. However, for everything else, the results should be fairly comparable to just a standard maxQuant execution. So let's have some look at the results. We'll first start with the results of the targeted database search. So these are the ProteoParc generated databases. And we can see that for bones, we've got a fairly decently sized bone proteome that we managed to identify, with also a fair amount of PSMs. For the dietary samples, we don't find a lot, which can be partially due to the nature of dietary samples. They are a difficult sample type to analyze, but the database size for these samples is also the smallest. And I'd like to emphasize that this is still very much a work in progress and that we're aiming to improve it. Now, for the enamel, we don't find a lot of proteins, but this makes sense because there are not a lot of proteins in enamel. We do find a lot of PSMs. So we also visualize the amount of peptides per protein. And be aware the y-axis for each of these material types is independent. And you can see that for enamel, we indeed find a lot of peptides per protein, indicating a very good coverage. Whereas for the bone, you see much wider distribution, and the dietary sample, again, we just don't find that much. Then, as for the running times, how much time did it actually take to analyze all these samples? We can see that the bone samples were the quickest, which makes sense because we told maxQuant to only look at the tryptic peptides, which drastically reduces the actual database size that the program has to deal with. Then the enamel, and lastly, the dietary samples. And that kind of confused us initially because that was the smallest database. And normally there should be a fairly straightforward relationship between database size and running times. We suspect that it took the longest because software like maxQuant, it tries to match every peak that you give it to a particular peptide. And if it can't find the right entry, it might kind of create a traffic jam, if you will, in terms of the analysis, where it just keeps trying to match it. And if the right entry is not in the database, it just might take unnecessarily long. So once again, it's very much a work in progress. I think this clearly indicates that we still have quite some work to do on the dietary sample side of things. But the running times for the bone and the enamel are probably representative. And you can see it still takes quite a fair amount of time, even though we have these targeted databases. Now, of course, the most interesting thing is to compare the amount of protein and PSMs that we have found from the targeted to the white databases, which I'll get to now. However, they are still running. It's been more than 3 weeks now since we've started the analysis, which we thought would be plenty of time. But the fact is that these UniProt-wide searches on maxQuant for the samples that we submitted is still ongoing. Although we can't make the comparison that we intended to do, this does provide some information that just the computational resources required to analyze these samples is so vast that it's probably just not feasible in many cases, which kind of emphasizes the need for these targeted databases. However, before we get too enthusiastic, we need to remind ourselves that there is still a lot of work to be done. So this work was done as part of one of the PAASTA committees for the database creation, and we are planning to expand these gene lists that we've curated. So far we've made one for bone, enamel, and dietary mixtures, and it's very clear that they still need a lot. The dietary especially still needs some work, but we are also currently working on some keratin databases, and we're planning to expand to other materials, including paint binders, eggshell, etc. We are also planning to create context-specific species lists. So right now, for the bone, we just downloaded on the level of mammals, which is quite large and also contains a lot of species that will probably not be present in the bone. So, for example, think of rodents, like something that could be quite easily excluded. So using the aid of the community of PAASTA, we are planning to create specific species lists for particular archaeological contexts to facilitate the easy generation of these targeted databases that are just the right size. Lastly, there's still some validation that we need to do. We've now done one simple test, and as you can see, there's still quite a bit of work to be done. We want to test more samples, but also more conditions. Think of how does the inclusion of multiple or different PTMs affect the running times and the amount of identified proteins with the different databases as well. And there lastly is an issue of gene annotation. So the way ProteoParc works is if you give the gene list, it then downloads all the records of the various taxa included in your taxonomic ID that have the same gene name. Yet we found that there are a variety of different genes that do not have the same name, or rather are not annotated with the same name across different taxa, even though the protein sequences are very similar. And this can give a false impression that you have included the records of a variety of species, even though they are actually not included in your database in the end. And this is, again, something that we will be working on and that we will keep you updated on in the future. And with that, I would like to give you the references of the papers we actually did, as well as thank our fellow committee members, Barath Meir and Laura Viñas Caron, for their work on this on these databases in this presentation. And then I'd like to thank you all for your attention.